Hi everyone, my name is Dr. Erin Dunn and I am an assistant professor at Harvard Medical School and run a research lab at the Massachusetts General Hospital. I'm really excited to be here with you today to tell you a little bit more about my work and work that's happening at the frontier of stress research. So my goals for today are to tell you a little bit more about work that's happening in three specific areas of research. The first is about how and why the timing of stress might matter. The second is about innovations that are now happening with respect to the measurement tools that researchers are using to try to measure stressful life events. And then the third speaks to the ways that researchers are trying to change the standards of practice in terms of how we actually do research. So let me tell you a little bit more about my work to give you some context for the presentation that I'm going to share. So the focus of my research laboratory is really organized around the who, the how, and the when related to stress. So our first goal is to really try to identify people who are at the highest risk for depression using genetic and other markers of vulnerability. The second thing that we try to do is to understand exactly how stress gets under the skin. So in other words, what are the pathways that the kinds of stressors we experience in the world today can literally get into our bodies and affect the functioning of the cells in our bodies and everything to shape our risk for long-term health outcomes. And then finally, we're also interested in trying to identify age stages in the course of the lifespan when our life experiences, including exposure to stress, might differentially matter. A lot of the work that I do is focused around depression, which for those of you who may not know is a major public health issue worldwide. So we know that depression is a disorder that's common throughout the lifespan. So about 1 out of every 5 people will experience an episode of depression at some point in their lives. We also know that depression is a disorder that disproportionately affects women. So during childhood, boys and girls experience similar levels of depression, but something happens during adolescence where girls start to outnumber boys by a ratio of 2 to 1, and that disparity persists throughout the rest of the lifespan. We also know that depression is associated with a host of negative consequences. Everything related to anxiety, missed work opportunities, recurrent episodes, side effects or medications, and I think in its most severest form, suicide. And because depression is so common, because it disproportionately affects large segments of the population and is associated with so many negative consequences, depression is currently the leading cause of disability worldwide. Now, my research group is really trying to find new ways to be able to prevent depression, which I think is really critical because depression is a disorder that strikes when people are young. And once it happens, it tends to be recurrent. So we know that about 20 to 40% of people who experience a major depressive disorder will have had their first onset before age 18. We also know that about 3 out of every 4 people with depression will experience a relapse at some point in their lives. So with data like this, I think the take home is that we need to really better understand the etiology or the cause of depression in order to prevent its occurrence, and to do that as early on as we possibly can in the course of the lifespan. Now, one of the things that we've been often focused on, which I know is of interest to you, is exposure to stressful life events. We know that these kinds of exposures are some of the biggest social determinants of depression. Now, this includes things like poverty, abuse and neglect, accidents or injuries, or even natural disasters and pandemics like what we're going through right now associated with COVID-19. We know that these stressors are associated with at least about a doubling in the risk of depression and other kinds of mental health and physical health related challenges. One of the things that I want to make clear up front is that you might have heard about childhood adversity, you might know a little bit about stress and traumas, And I just want to use this slide to give people a little bit of a better sense about what all of these different terms mean. And I think the key thing to take away from this slide is that we often use these different terms to capture different aspects, and to be keeping that in mind as you think about the work that I'm presenting in terms of this presentation. So what I want to do now is to transition into telling you more about work that's happening in this first frontier, and that is related to how and why the timing of stress might matter. So one of the things that my research group has really been trying to get a better handle on is exactly how childhood adversity in particular might increase people's subsequent risk for developing depression. And in this way, there's been a number of different theories that researchers and scientists have been trying to test. One is a basic exposure model. And this model simply says that people who are exposed to adversity and different kinds of stressors have an increased risk of depression or other adverse mental health outcomes compared to people who are unexposed. There's also an accumulation model that people have been testing, and this focuses on the number of different adversity exposures or the duration of those exposures. And here I'm presenting a basic dose-response relationship where the number of exposures to adversity is proportionally associated with people's risk for depression. A third model that people often talk about is a recency model, and this focuses on the time since the onset of exposure. And here we see that the risk for depression is greatest shortly after people have been exposed to stress and adversity, but that effect dissipates over time. And then the final model that my group is really in particular interested in is this idea of sensitive periods. In other words, that there might be age stages in the course of the lifespan when exposure to stress and adversity is even more potent in terms of conferring risk for depression. One of the reasons why my research group and other research groups are so interested in trying to understand which of these different theoretical models might be driving risk for depression is because it's going to have profound implications for how we intervene. So if the data are more consistent with an exposure model, that suggests that we can intervene at any time. And our goal should therefore try to be to identify people who've been exposed. If it's more of an accumulation model, it suggests that we want to try to intervene early before people start accruing a greater number of exposures to stress and adversity. If it's a recency model, it suggests that we want to try to intervene quickly. And if it's a sensitive period model, it suggests that we want to intervene during very specific stages in development, either before or during these sensitive periods in development. In this way, I think about sensitive periods as being both high-risk periods or windows of vulnerability when exposure to stress and adversity is even more potent, but also high-reward periods or windows of opportunity when enriching life experiences might also be even more beneficial. So one of the first studies that my research group did to try to understand and see if there might be sensitive periods in development came from analyses that we did using a data set called the National Longitudinal Study of Adolescent to Adult Health. And what we did in this study was we looked at the association between age at exposure to two different types of childhood adversity, specifically physical abuse and sexual abuse. And we looked at how the age at first exposure was associated with an increased risk of depression. And what we found is that generally across the board, people who reported being exposed to physical abuse had an increased odds of developing depression compared to people who were never exposed. But when we dug a little bit deeper in terms of the age at exposure, we found that people who reported being exposed to physical abuse for the first time during preschool, when they were 3 to 5 years of age, had an increased risk of experiencing depression compared to people who were first exposed during adolescence. And similarly, for sexual abuse, we also found similar findings where generally across the board, people who were exposed had an increased risk of depression, but here too we also found timing differences. Here it suggests that the period of latency, or about ages 6 to 8, was the time period when children were more likely to experience depression as a result of being exposed to sexual abuse. One of the things that we've also started to look at is the process of epigenetics, and in particular a type of epigenetic mechanism called DNA methylation. So epigenetics is a mechanism for controlling when our genes are turned on and turned off. Every cell in our body has an identical genome or sequence of DNA. This is true whether it's a muscle cell, a liver cell, or a brain cell. But what's really remarkable is that all of these cells have different structures and functions. And one of the things that makes this possible is that because within each of our cells, the DNA can be tagged by tiny chemicals that change how the genes are expressed. These chemical modifications are known as epigenetic marks. And you can think about epi as the Greek word for meaning above, or above the genome. These epigenetic marks can cause some genes to be turned on and others to be turned off. One of the most commonly studied types of epigenetic modification is called DNA methylation. Recent evidence from a number of different studies suggests that these DNA methylation marks can be influenced by our life experiences and things that are happening within our environment, including exposure to stress. One of the things that my research group has done is to try to look at how the timing of exposure to adversity shapes these DNA methylation signatures. So this is data that we analyzed from a cohort study out of Bristol, England, called the Avon Longitudinal Study of Parents and Children. And in this data set, parents reported whether their children were exposed to a range of different types of adversities in the first 7 years of life. So at each of these different time periods that I'm showing here, parents were reporting about whether or not their child had been exposed to one of these different types of adversities. And we looked at the relationship between the timing of exposure to adversity and DNA methylation at about 500,000 different sites within the genome. And what we wondered about and what we sought to actually test is what theoretical model that I just shared with you best explains variability. In other words, which of these theoretical models seem to be most likely to explain the association between exposure to adversity and DNA methylation across the genome. So what I'm showing here in this plot is called a Manhattan plot. So on the x-axis is the chromosome, and on the y-axis is a statistical measure of the degree of association between each individual CpG site or DNA methylation mark and exposure to adversity. What we found is 38 significant CpG sites or DNA methylation sites that were distributed throughout the genome. In other words, these were DNA methylation sites that showed evidence of having been influenced by exposure to adversity. When we dug into these findings a little bit more, we found some really interesting results. So what we ended up finding is that of the 38 CpG sites that were associated with exposure to adversity, more than half were identified as being influenced by exposure to adversity in very early childhood. So in other words, when we looked at whether the results were driven by one of these theoretical models, there was very clear evidence showing up for the sensitive period model more than any other model as driving these DNA methylation patterns we were finding. I now want to tell you a little bit more about work that's happening to try to identify new measurement tools to record stressful life events. So one of the things that's become really clear is that the current tools we have available as scientists to identify people who are at high risk for mental health and brain health related challenges like depression is really limited. So what I'm trying to show in this table are some of the commonly used measures to record and to characterize people who might be at high risk, and that's shown in the left-hand column. And then in the top row, what I'm showing is the different kinds of scientific evidence that we can use to support these different measures. So in other words, whether they're reliable or whether we get the same answers to these different kinds of measures when we look at them repeatedly. Are they valid? Do they tell us what we want to know about that particular type of risk? Are they non-invasive? In other words, how cumbersome is it for us to be able to access that kind of information? Is it easy to access in terms of availability in both children and in adults and with respect to different kinds of populations worldwide? And then finally, the last column gives more information about whether it might be inexpensive to actually analyze. And what I think is most striking is that if you look at this table, what you can see pretty clearly is that a lot of the tools we have right now just don't fit the bill. They have some pretty serious limitations with respect to all of these different domains that make it really hard for us to be able, as scientists, to identify people who are exposed not only to adversity, but might be at high risk as a result of family history or genetic markers or things of this sort. So to give you a better sense of this, so one of the things we know is that commonly used tools to measure exposure to stress and adversity in particular have very serious limitations. So one of the strategies that I think is most often used is retrospective reporting, where people in adulthood are asked to self-report whether or not they've been exposed to a range of different kinds of stressors during their lifetime. And as you might imagine, this kind of approach to measurement has a number of different problems. So for example, retrospective reports are susceptible to bias in terms of our memory, especially when people are asked to record when specifically these life stressors may have occurred. We also know that people might be reluctant to talk about these experiences and might be less likely to disclose this based on the relationship they might have formed with the interviewer who was completing the scientific interview. We also are using prospective measures within scientific research where we're asking people, in particular mothers or other caregivers, to report in real time whether children are exposed to different kinds of life stressors and traumas. But as you might imagine, there's also a number of different challenges with respect to prospective methods. So parents might, for example, be embarrassed or reluctant to implicate themselves in their child's exposure to different types of stressors. Parents might also be unaware that their children were exposed to certain kinds of stressors. And I think when we look at this from the perspective of when we have data actually available from both children and parents, what's so striking is that we often see very low levels of agreement between parents and their children. Another commonly used method has been to take official reports, such as health or social service records, to characterize child maltreatment, for example. But we know that these are often problematic in that they fail to detect all cases and really are only detecting a very small fraction of cases. So it's been born from these frustrations that my research group has been wondering about whether children's shed teeth could serve as a new biomarker of exposure to life stress. We recently published a paper in the journal Biological Psychiatry that proposes this teeth model to characterize teeth as potential new tools to measure early life adversity and people's subsequent risk for mental health related problems. So I want to tell you a little bit more about what we've been doing and what this paper describes in terms of this teeth conceptual model. So teeth, here's the timeline of tooth formation. So a little bit of a tooth primer. So there's 20 primary teeth which form prenatally through the first 3 years of life. So our earliest forming teeth, our primary teeth or our baby teeth, start forming in utero. Those primary teeth are then replaced by permanent teeth, which form postnatally up through about mid-adolescence. Teeth are really remarkable resources in that they record the timing of their incremental growth. So here I'm showing what a cartoon of a tooth. So a tooth is comprised of a crown and a root. The enamel and dentin are the parts that comprise the crown. And the enamel is the white outermost part of our tooth crown. So during development, there are cells called ameloblasts that are laying down the enamel that exists within that outermost part of our crown. And the way that ameloblasts work is that they operate in a process where they're essentially laying down the layers of enamel in these incremental patterns, very much like a circadian pattern that's occurring daily across time. What's remarkable is that this developmental process is permanently recorded in the tooth, much like the rings in a tree that mark its age. Teeth also record insults or disruptions that occur during their development. So this can happen both on a low resolution time scale and a high resolution time scale. So most of the stressors or disruptions that people have studied include things like illness and malnutrition and heavy metals. So you can see on the low resolution time scale that I'm showing here on the right that there are only certain teeth are involved in these disruptions here. And this is the way that we often talk about it, that every tooth can tell its own story. So the fact that these marks or these hypoplasias are concentrated to certain teeth tells us a little bit about when in development these stressors might have occurred. But building on the idea that teeth also record their incremental growth in the way that trees record their incremental growth, you can also see evidence of developmental disruptions on a high resolution time scale. So in this way, teeth can tell us not just whether a stressor occurred, but specifically when. And this happens through the occurrence of these growth lines and in particular, these stress lines that can be seen within each tooth. I think what's really remarkable is that teeth could provide enormous and unique opportunities for primary prevention of depression and other brain health related challenges. So you might remember from the beginning of my presentation that I said that about 20 to 40% of people who experience depression will have had their first onset by age 18 or about age 20. The way that I think about it is that every time a tooth is lost represents an opportunity to potentially intervene. So the first opportunity happens when teeth are naturally shed. So this happens around ages 5 and 6, when teeth naturally fall out. This could be an opportunity to think about ways to capture teeth and then use those teeth to make estimates and predictions about children who might have experienced early life adversity and therefore are at subsequent risk of experiencing depression or other kinds of adverse mental health related outcomes. The second opportunity comes from orthodontic work. So I think the latest data suggests that about 15% of people in the United States will have had one or more teeth extracted for orthodontic work during adolescence. Oftentimes what happens is that a lot of those teeth get thrown out. But imagine if we could think about using teeth instead, again, to be able to use as a new tool to be able to capture exposure to stress and different kinds of stressors. The third opportunity comes with the time period of early adulthood and emerging adulthood, which we know that just like adolescence is a time period when many mental health related problems start to first emerge. Wisdom tooth removal surgeries are pretty common at this time point. So here again, instead of throwing away those teeth, we might think about this as a time period when we can instead study teeth and see if we might see evidence of people's exposure to a range of different types of stressors and traumas. So building from these possibilities, I've decided that I want to become the science tooth fairy who collects and studies teeth. We've been undertaking a number of different research studies to try to investigate the relationship between a number of different types of exposures to stress and trauma and how they are recorded in teeth. One of the ways that we've been doing this is to try to collect children's baby teeth. And so what I'm showing here is an example of a tool that we've been using for recruitment purposes. So my research group recently wrote a children's book that describes the collection that we use for teeth and what we do in my research lab to try to understand the kinds of things that might be recorded in children's teeth, and to use this as a tool to essentially get kids excited about wanting to send their teeth to the Science Tooth Fairy. For anyone who's interested in learning more about this work and what we are sinking our teeth into, I definitely encourage you to visit our website, teethforscience.com. So now in this last section of my talk, I want to tell you a little bit more about work that's happening to really change the way that we do scientific research. So I think that we desperately need breakthrough discoveries in the field of brain health. We know that brain health problems affect about 1 out of every 3 people worldwide. This is true whether it's depression, anxiety, Alzheimer's, PTSD, Parkinson's, and so forth. But there's a limited understanding of the causes of brain disease. And this is problematic for 3 reasons. The lack of knowledge about the causes of brain disease limits our ability to treat brain diseases once they happen, to prevent them from occurring, and from figuring out ways to promote brain health across the lifespan. I think for this reason, discovering the basis of brain health may be one of the most important medical challenges of our time. I think what's amazing is that these needs exist worldwide and nationwide, but they're really concentrated in certain parts of the country, and in Florida in particular. So if you look at the data from Florida, you can see that they rank 48th out of the 50 states for adults who have mental health related challenges who don't receive mental health care. They rank high with respect to the number of available mental health providers per capita. They rank very low with respect to spending on mental health support. And they're also ranked quite high in terms of the number of people who have major depression. So we, as a research group, have decided to launch this project called the Brain Health Initiative, the Florida Gulf Coast and beyond. And our overarching goal is to identify, develop, and implement interventions aimed at optimizing brain health and performance throughout the lifespan. So to accomplish this goal, we're working to identify the factors that shape brain health, including the role of genes and lifestyle factors, and aspects of the social and physical environment. We then want to use this information to then identify and understand the effects of different types of interventions to promote brain health and reduce the risk of brain-related diseases, and also determine how these factors unfold across development so that we can efficiently and effectively target interventions to when they're going to have long-lasting impacts. This project is led by a multidisciplinary team of scientists and clinicians. So I'm the director of research for this project, and I'm really excited to be involved with it, including with efforts from Heidi Hanna, who so graciously has invited me to be part of this Global Stress Summit. We're also working with a range of different local, national, and global collaborators who span a range of different disciplines. So I'm an epidemiologist by background. I also have done work in the field of psychiatry. We have folks that come from a range of different disciplines. And I think what's most exciting about this is that I think in order to answer the challenges that come with understanding brain health, we have to really be thinking in this multidisciplinary way. And so it's so exciting that our team is comprised of so many different multidisciplinary scientists. In terms of how we're working to achieve these really ambitious goals, so there's kind of 3 main pillars of this project. So the first is to engage in a longitudinal observational study where we're working to identify the predictors of brain health among a multi-generational cohort. The second arm of our project is to launch a living laboratory where we can test the impact of novel interventions to improve brain health and enhance performance. So the idea here is to not just do observational studies alone and not just do clinical trials on their own, but to really marry the two in a way that quite hasn't been done before. And also to bring these interventions to where people are, whether that's where people live, where they work, where they play, where they're learning with respect to schools, and so on and so forth. And then the third pillar of this project, which I think is also unique, is that we're engaged in a collective impact model. So we recognize that in order to address the enormous challenge of brain health, we have to figure out how to engage with multiple stakeholders, not just scientists, not just clinicians, but really it's going to take everyone. So this is one of the reasons why we've been working with stakeholders or our brain health champions across the community to help us increase the sustainability and long-term success of this project. So I want to end this presentation by making a couple of closing remarks. I know that we've talked about stress, we've talked about adversity and trauma, and these can be really hard things to hear about, especially right now as we're living through this COVID-19 pandemic. But a key thing that I want people to really take away from this is to not lose sight of hope. Because what we know as a scientific community is that most people, despite experiencing stress and adversity, do okay. So you can see, for example, that on the left, I'm showing that there's lots of individual differences. We know that even people who've experienced high levels of exposure to stress and adversity can still do well. And in fact, that we know that about 2/3 of people who have experienced stress and adversity as children don't actually develop mental health related outcomes, adverse mental health related outcomes. So in fact, resilience is actually the norm, not the exception. I think this begs the question of, well, how do we actually go about trying to promote resilience and resilient related processes and outcomes? And I think here's where it's really critical to think about ways that we can identify the buffers, in other words, those protective factors that can offset risk for poor health outcomes, and how we can do this for everybody, and in particular for people who've been exposed to early life adversity and different kinds of stressors. I think we should be thinking about these opportunities for identifying buffers across different levels of analysis. So in other words, what are individual behaviors? What are genetic factors? How can we think about social relationships, our living conditions, neighborhoods and communities, the way that our institutions are structured, as well as the social and economic policies that drive how our country is run. So I want everyone to be thinking about opportunities to buffer and to create safety nets at all of these different facets. So from the micro to the macro level structures of society. I also want to share with you what we are referring to as our 10 pillars of brain health that we talk about in the Brain Health Initiative. And these are 10 things that are really lifestyle factors and behaviors that people can do to try to buffer the effects of exposure to stress. And I think it's really key to emphasize these now as we're going through this worldwide pandemic. So as to the extent that people can, and I know that it's really difficult to do this now, but try to think about ways that you can boost your stress resilience through things like meditation, for example. Eat well, try to get those vegetables in, try to not drink sugar sweetened beverages, get out there and move, whether it's just going for a walk, doing some cardio exercises in your home, get good sleep where you can, stay connected. So we're physically distancing, but we shouldn't be socially distant. So reach out to those people that you haven't connected with in a while and say hi. Tell them when you feel like you need to hear from people and work to try to stay socially connected. There's lots of things that we can do to keep our brains active in terms of cognitive stimulation, puzzles, all sorts of museums are offering free tours and other things of this sort. So I definitely encourage you to check those out. Stay connected with your emotional well-being. Reach out for help from professionals when you feel like you need it. Try to find meaning and purpose in every part of your day-to-day life, whether that's realizing where you feel gratitude, whether that's in terms of trying to identify ways that you can change the work that you're doing to have greater meaning. There's lots of, I think, ways to derive meaning and purpose, even in the face of everything that is going on in the world right now. Be sure to also keep good general health to the extent that you can, and recognize opportunities for you to make a positive impact. And I think here the two biggest ways that we can have a positive impact right now are in trying to do what we can within our local communities to make a difference. That might mean donating blood. It might mean staying at home. So a lot of the work that folks are really emphasizing now in epidemiology and public health is to stay at home, and that will do the biggest, that will have the biggest impact that we know of to really shift the curve with respect to this COVID-19 pandemic. So let me end by saying that if you like a lot of what I've talked about today, I definitely encourage you to check out my research blog called Erin Dunn on our website, thedunlab.com/blog. We include information here focusing on a range of different scientific topics, tips and how-tos, and also sources where people in the research and clinical communities can find sources of inspiration. And then I'll, of course, like to end by thanking members of my research group and all of the sources of funding that support my work, and point out my email for folks if you've got questions and want to follow up, in my lab website and social media if you are interested in following the work that's coming out from my group. So with that, I'll bring this presentation to a close. Thank you for everyone who tuned in. Feel free to reach out to me if you have any questions, and I'd be happy to do my best to try to answer them. Stay safe, stay healthy, and be well. Thank you so much.